I'm a paleolimnologist by training. Most of my career I've studied lake sediments, and some of these lakes were in coastal regions, but recently I've made the plunge into the marine realm. So in the actual coastal sediments on the marine side. Today I'd like to just present the type of human traces that we can find in these coastal sediments, and I'll try and use examples from North America in keeping with today's focus, so for this workshop. By now we've all heard about the Great Acceleration and all the facets of global change that show a steep increase around the mid-20th century. And last year, Jouffret and his colleagues published a paper in the journal One Earth, where they took the same idea of the Great Acceleration and applied it to the marine environment. So they coined the term the blue acceleration to show the similarity of the increases in global change in the marine realm. So the similarity between the terrestrial acceleration and the marine acceleration of human impacts. But this one starts a little bit later. So instead of the mid-20th century, we're more in the early 21st century for the marine realm. The authors point out that claiming marine resources and space is not new to humanity, of course, but the extent, intensity, and diversity of today's aspirations are unprecedented. So inevitably, these familiar curves alert us to the increasing threats to the marine environment that we pose, including, of course, the coastal areas. So over various timescales and different parts of the world, our species has become a major factor of coastal environmental modification. As a matter of fact, coastal zones are the world's most densely populated regions. So inevitably, we're going to have a strong impact. We're able to completely transform coastal ecosystems, as these images of Manhattan illustrate. And we're also doing it increasingly rapidly and intensely. So coasts, like lakes, are complex biophysical systems at the interface of land, air, and water. And of course, humans have been present, as I said, in coastal regions for a very long time, for millennia. And the dissemination of human society imposes further complexity to these coastal systems. So to understand human impacts over time in these regions is important in order to better understand how the current situation is affecting coastal ecosystems in the complex of a rapidly changing environment. So this figure here by Hay and Silliman, published in 2019, shows the synergistic effects of the climate change and the local human activities on coastal ecosystems. So in A, you have no climate situation where you have no climate change, so pre-Anthropocene, and a low population density. So this would illustrate, for example, our baseline conditions. And in B, we just add climate change to this low population density. So we're getting only a climatic signal linked to rising CO2 levels and rising air temperatures, for example, that are having an effect on the water, the aquatic ecosystem. So we're having higher water temperatures that are leading to more stratified water column, the thermal expansion of the water, sea level rise, ice melt, precipitation changes, etc. And in C, we have both climate change and high population density, where we now see a synergistic effect of both these changes on the aquatic ecosystem. So on top of rising CO2, on top of rising temperatures, we have local and regional impacts of pollution, such as wastewater that's coming into the ecosystem. We have aquaculture, we have nutrients coming in from the terrestrial agriculture, we have overexploitation of marine resources, et cetera, et cetera. So all these impacts have been identified by syndromes of global change by Newton and collaborators, among others. So these syndromes of global change are problems, challenges related to global change. And they include, for example, the biodiversity syndrome, which is from stress and overexploitation of biotic resources, the introduction of invasive and exotic species that lead to changes in the food web and regime changes, etc. Another example is the pollution and contamination syndrome. So these obviously are from industrial sources, agricultural sources, accidents like oil spills, and underwater noise that we create, the marine litter. And finally, another example is coastal land use syndrome. And this is the destruction of the coastal ecosystems, such as the coastal forests, the mangroves, the wetlands, the salt marshes, and also the overexploitation of sand. So the destruction of beaches just by the extraction of sand that we need for the concrete. So all the buildings that we're creating. So one of as with lakes or the deep ocean, all of these coastal environmental changes and perturbations are being recorded in sediment archives. And one of the biggest challenges of doing paleo research in coastal zones is recovering cores that are suitable for this type of study. So cores that have good sedimentary accumulation that's constant and minimally interrupted. So we're dealing with zones that are usually highly dynamic with strong currents that can transport sediment for significant distances. And so finding the right location to core is somewhat of a challenge in coastal areas. But it's definitely possible. And here we have two examples of cores that are from coastal regions. So the one on the left is a sediment core that was taken in the Great Blue Hole off the coast of Belize. So the Great Blue Hole is a sinkhole. So it's actually like a lake in the ocean. This particular one is about 125 meters deep. So it's very deep. And as you can see, with the naked eye, it's possible to see the lamination, so the deposition of the sediment, and it looks to be pretty much undisturbed. So this would be a very good sediment archive to study. And the other example that we have on this slide on the right is an example of a box core that was taken last summer by a colleague of mine on the North Shore region of the Gulf of St. Lawrence in Quebec province. So you can see the box core and the two tubes that are stuck into it that we're actually going to use for the study. This one shows less of stratification to the naked eye, but it still appears to have some stratification, some changes that are visible. So this will probably provide us with a wealth of information on the past variations of this region, which I'm going to talk about a little bit more later on. So exactly what kind of human traces can we find in the sedimentary records of coastal zones? To be honest, it resembles a lot what we can find in lake sediments. So one of the most obvious impacts is the flux of terrestrial sediments to the aquatic system. So this is usually one of the first human traces that we find in lake ecosystems, as we found in our study that was published with the previous working group. So there's human activity in a catchment where we have perhaps deforestation. And so the sediment, the terrestrial sediment, is more easily transferred to the aquatic ecosystem, and it's the same in coastal zones. So, for example, here we have a study by Savitsky and collaborators who evaluated the flux of terrestrial sediments to coasts in the pre-Anthropocene and compared it with what it should be today. So you have different zones where you'll have either an increase or a decrease in the flux of terrestrial sediments to the coast. So that'll depend on what human activity is going on in the catchment. So for example, if there is a large dam that's built on a very large river, it's going to slow down the flux of terrestrial sediments to the coast. So it's one kind of human trace that we can find in our sediment record. It'll also be easy to see with the sediment changes in the sedimentation rate, with the lithology, the type of sediment that's being deposited, the grain size, the carbon content, etc. So there are a lot of impacts, a lot of human traces that we can find in the sediment that's linked to the flux of terrestrial sediments. Another manifestation of anthropogenic activities on the coasts is via the biodiversity, the effects that the humans have on biodiversity. Of course, as I mentioned before, there's the introduction of species. There's also the extirpation of species with overexploitation. We have deliberate introductions, accidental introductions that are often linked to maritime activities. So one good example is the zebra mussel in the St. Lawrence system. And once a species is introduced into an aquatic system, it's really, really hard to get rid of, as you can imagine. So the graph that you see here is a study by Lutz et al. Published in 2006, where they went through the historical record. So this is not actually based on the sediment record, but the historical record of biodiversity in coastal zones. And now a lot of these zones are actually located in North America. So here in E, you have the different zones that were studied and the present state of relative biodiversity, abundance of biodiversity in 6 taxonomic groups. So a lot of the very degraded systems are in Europe, so the Adriatic and the Wadden Sea, the Baltic Sea. And then as you move to North America, the systems are not as degraded, but they're getting there if you look at the trends. We still have our hockey stick trend that's present in most of the systems that were recorded. So how does this change in biodiversity manifest itself in the sedimentary record? There are a lot of indicators here also that we can use. There's microfossils, there's pigments, there's sedimentary DNA that is becoming one of the go-to proxies for evaluating coastal biodiversity. And so some of the coastal microfossils that we have access to, you can see that there are a lot of them, they're very diverse. So most of these organisms, which are either zooplankton, zoobenthos, phytoplankton, we have spores, we have pollen. These organisms are sensitive to temperature and salinity. And some are also sensitive to dissolved oxygen, to pH variation, and even to nutrient content. So on top of being able to give us information on biodiversity, they can also inform us on the variations in these environmental variables based on the changes in the assemblage composition of these remains. Another anthropogenic trace, another human trace in coastal records, of course, is pollution by metals, increase in nutrients and pesticides that come from mostly come from terrestrial use. I wasn't able to find a really good example from North America for metal pollution, but there are a lot of them in Asia. And so this is sedimentary evidence of late Holocene human activity in the Pearl River Delta in China over a 4,000-year record. So you can see the increase in copper, increase in iron and lead, and this is also reflected in the magnetic susceptibility of the sediments. So this is what I like about this example is that it shows us pre-Anthropocene conditions, so the baseline conditions, and then the intensity of the increase in the human footprint. And then an example that I found for Eastern North America is by Cooper and collaborators in 2004 in North Carolina on estuaries. So we have an increase in nutrients and an increase in nitrogen and in sulfur, as you can see in the 20th century. So it's quite evident. So this also shows us the pre-colonial signal, I would say, not the pre-human signal, because of course there were Native Americans in this region before 1600. But it corresponds, the base of the record corresponds neatly with the colonial times. So another human trace that we can find in coastal sediments is coastal eutrophication, just as we find in lake sediments, and also hypoxia due to eutrophication. I was actually reading the news this morning, and in Turkey, at the moment, they're caught with they're facing a problem with what they call sea snot, and this is the production of extracellular. I forget the term, but anyway, it's produced by diatoms, and it's really like snot that floats on the river, and this is due to eutrophication, to the increase in nutrient load. And so it's a good example of a coastal case of eutrophication right here, right now. But this figure is from Good Day and collaborators in 2009. And it shows us eutrophication near the Mississippi Delta, so the southern United States. And you can see the increase in organic accumulation, and it's very nicely linked to increase in fertilizer and increase in land drainage and all of the other variables that we talked about earlier. And on the right, you have this eutrophication that is visible in changes in ostracod assemblages. So you can either measure it directly in the sediment via geochemical proxies, or you can use the biological proxies to trace this eutrophication and hypoxia in coastal sediments. There's also global change that we can trace. So we've talked mostly about the direct regional changes and human traces in the coastal zones, but global change is also very much present via ocean acidification and warming. So on the left here, you have a nice correlation between the increase in the release of CO2 into the atmosphere, which is the pink curve, and the acidification of ocean water in the orange curve. So these are inferred trends, so based on modeling, but you can see that the two are inversely correlated very nicely. Another global change that we're facing is the warming of the oceans. And at our latitudes in Canada, that means the melting of sea ice, whether it's seasonal sea ice or permanent sea ice. And there are very strong indicators of sea ice, so the biomarkers of sea ice in either diatoms or forams or ostracods and dinoflagellates. So some of these groups have species that are specifically adapted to sea ice that grow only on sea ice. And so it's possible to infer the fluctuations in the coverage of sea ice in coastal zones using these. So my work during the next 5 years is going to take place in the region of Sept-Îles, which you can see is located in the Gulf of St. Lawrence in the northwestern Atlantic. Actually, Canada has by far the largest coastline of any nation on Earth, and it's surprising that there hasn't been that much research done on coastal system sediment archives. And so I found a really great spot for studying coastal systems, because Sept-Îles has a very nice bay, which you can see in the figure here on your right. So this bay is about 45 square kilometers in area. It has a maximum depth of about 80 meters. And you can see here, the blue outline is industrial zone. And the red here is the city. So the city of Sept-Îles has about 30,000 inhabitants, which is not very many. For Quebec, it's an average sized city. And the interesting thing, though, is that even if there aren't that many inhabitants here, there's a lot going on. There's a lot of industrial activity. One of the largest, if not the largest, aluminum smelter is located here on Black Point, Pointe Noire. There's a deep water port. There's a lot of, you see the railway here, there's a lot of mineral transiting through the Sept-Îles area. So the mineral is brought in from the mines in the north, and it's transferred onto boats that ship out to all corners of the world. So that means that we have a lot of impact of human traces in this zone. So as you can see in the middle photo here, there's often accidents, as I was mentioning. So this is an oil spill that happened a few years ago. There's also a thriving fishing industry. So there's a lot of seafood fishing, shrimps and crab. And also before the pandemic, the development of tourism was increasing very fast. So you can see here the large cruise ships are stopping in Sept-Îles. So since it's a deep water port, there's no problem. We can have huge cruise ships stopping here. So this means that between the city and the industrial zone that's adjacent to the city and the other industrial zone on the other side of the bay, we have a hotspot here of human impact. So you can imagine all the ships that are waiting in the bay to load or unload, well, it's mostly loading, the minerals. They just keep their engines on, so there's a lot of pollution being created. And luckily, the port administration is very sensitive to their impact on the environment, and they're very interested in the research that we do, and they're funding a large portion of it. So we have the means to make a very, very in-depth evaluation of human impacts on this zone. So the next time I present to the working group, I hope to have results to show you. We're evidently going to study a number of proxies, geochemical and biological. And so I hope to be able to present these to you very soon. Thank you.
Thank you, Emilie, for this nice overview of human traces in coastal sediments. Do we have any questions for Emilie? If so, just speak up or raise your hand or write in the chat as we did before. I had one but it's probably easy. On the Cooper paper, where you showed the proxies about, there was nitrogen increase and sulfur increase.
Yeah, in North Carolina.
I'm just wondering, because I'm not aware of this paper, did they interpret the sulfur as a proxy for hypoxia, or just input of sulfur from the land?
I think so, yeah. Yeah. If I remember correctly, I read a bunch of papers in the past 2 days to prepare my presentation, but yeah.
There's Jesse
It can be both, but it's a good example of a proxy for hypoxia. Yeah.
Good. Jesse raised his hand, and then we have also a question in the chat already. So Jesse, please.
Yep, great. Thanks, Emily. Really interesting talk.
Thanks.
I haven't done a lot of coring or any coring in coastal systems before, so I'm just curious how you decide where to core and you know, how wide spread is the signals from like an oil spill or something like that.
Yeah, it's a real challenge, as I was saying. It's easy if you find a bay like Sept-Îles, because I don't know if you remember the map. So it's almost like an enclosed bay, because you have all these islands at the mouth of the bay. So I think most of the sediment will stay inside the bay. So it's like a big lake. But we have people, other researchers that are studying the currents, marine currents. And so my reflex as a paleolimnologist would be to core the deepest spot in the bay. But we do have cores from different spots. And that's one of the things that I'm planning on looking into, is how does the signal vary from one spot to another. So we have a core from the 80-meter depth, so the deepest part of the bay. And we have another core, which is also from the hot spot, but at a 40-meter depth. So I'm going to try and see if the 80-meter core really gives us more information, or if they both show the same thing. But on a normal coast where you wouldn't have like a protected area, I think it's probably really hard, and it's probably why there hasn't been that much paleo work done in the coasts in Canada. So I really don't have the definite answer how to choose the best spot, but I'm hoping to get better at it and learn from my colleagues who are oceanographers and who have been coring these spots for a much longer time than I have. So when I have more information, I'll share it with you guys for sure.
Thanks.
Okay, then we have a question in the chat. Which proxy, biological or geochemical, is better for studying eutrophication, and why, or which one do you prefer?
Well, I would say the best approach is the multi-proxy approach, of course. So you have to use both geochemical and biological markers to really get a good idea, a good picture of the eutrophication. Personally, my favorite biomarker is diatoms. But I really think that the combination of different biological markers and also the geochemical markers, so the carbon to nitrogen ratio for the provenance of the organic carbon, and the other proxies, so the changes in the sediment, the nature of the sediment, if you like. Those are all really good markers for eutrophication. Also, there's, I think you can use like molecular proxies. Those are more and more common. So if you have access to sedimentary DNA, You can use those, and those are really, the sedimentary DNA is an excellent biomarker for changes in biodiversity. So I think a lot of that is being developed these days. And of course, yeah, my final answer is you really have to use as many proxies as you can for any signal that you're looking for, whether it be eutrophication or climate change or pollution.
There's an additional question. What is the temporal depth that you plan to study?
Hopefully I have a core in my fridge that spans the last 2,000 years by my estimates. We don't have any radiocarbon dates yet. But that's about the time span that I plan to study. So I would give us an indication of baseline conditions. Of course, there have been Native Americans in these parts for many thousands of years. So we would probably get a signal of their activity. And then the city of Sept-Îles is a very new one. It was only founded in 1950. So it's the case in a lot of Canadian regions where the urban zones are very new, very recent. But there has been activity in the Sept-Îles area for about 200 years. So a lot of fishing. There was even whale oil industry. So the Norwegians had an outpost there and were fishing or hunting whale and collecting the oil. So it's different timescales. I really like comparing the recent signals. So the last 50 years with the last 100 years and then with the last 1,000 years. I find that it gives you a really good perspective on what are the major influences on the changes that you can see in your archive, in your record.